The overall goal of this procedure is to prepare Drosophila pupae for live cell imaging of dividing thoracic sensory organ progenitor, or SOP, cells during metamorphosis. This is accomplished by first setting up the appropriate genetic cross to obtain progeny pupae for live cell imaging. The second step of the procedure is to collect Drosophila pupae at the appropriate stage to visualize asymmetrically dividing neural progenitor cells. The third step of the procedure is to remove the pupal case in order to mount the pupa for imaging. The final step of the procedure is to mount the pupa between slide and cover slip and proceed to live cell imaging. Ultimately, results can be obtained that show the dynamics of fluorescently tagged proteins during asymmetric cell division and differentiation of wild-type or mutant sensory organ cells through either epifluorescence or confocal microscopy.
The main advantage of this technique over existing methods like immunohistochemistry is that protein dynamics can be followed in real time and over the course of several hours in an intact pupa.
From a cross of the desired Gal4 line and GFP-tagged fusion protein under UAS control, identify pupae that have undergone pupariation within the hour. These pupae will possess characteristic pupil morphology but lack pigmentation. Selected pupae can either be transferred to a fresh file or can be marked on the cross file to remember which ones were white 18 hours later. When the pupae reaches the appropriate stage, place a piece of double-sided tape onto a microscope slide and adhere the pupal case to the slide, ventral side down. Under the dissecting microscope, identify and grasp the edge of the operculum using a pair of forceps and gently lift it off, revealing the head of the immature fly. Being careful not to puncture the body of the fly, gently tear the side of the pupal case and remove it completely to reveal the thorax.